Great. Hi, everyone, and welcome to our fourth seminar of our Animal Behavior Live seminar series for 2022. So today we have Dr. Rohini Balakrishnan and as our speaker. So before we begin, just a quick reminder that you can ask questions in the YouTube chat at any time, and then we will just relay them to our speaker during the question and answer period. So a little bit into Rohini's background. So she is currently a senior professor at the Center for Ecological Sciences at the Indian Institute of Science in Bangalore, India. She received her PhD in behavioral genetics at the Tata Institute of Fundamental Research in Mumbai, India, under the supervision of Veronica Rodriguez. She then completed 2 postdoctoral positions, one at McGill University in Canada, and then another at the University of Erlangen, Germany. And that focused on behavioral ecology and acoustic communication. So her research currently focuses on understanding a variety of animal behaviors and communication systems, including predator-prey interactions, acoustic communication, and mate selection. And so her talk today will be entitled Song, Flight, and Death, Benefits and Costs of Acoustic Communication in Crickets and Katydids. So thank you, Rohini, for joining us today, and you can go ahead with your talk.
So thanks very much to Alexei and Amanda and the rest of the organizers for inviting me to give this talk. And I think without further ado, I will move on to my talk. Right, so I'm a behavioral ecologist, and I'm interested in acoustic communication and acoustic signaling. And I'm interested in pretty much every aspect of them, from their diversity, their perception, functions in an ecological and evolutionary context. And I have been studying these over the past 30 years, largely using crickets as a study system. And just to sort of put crickets in context, they're jumping insects of the order Orthoptera, which is 2 major groups, the crickets or the Encephera, the grasshoppers of the Califera. Very large numbers of species of crickets produce sound, and they typically produce them by wing stridulation, that is by rubbing their wings together. And their ears tend to be on their legs. You can see this little white spot over here. That's an ear. Grasshoppers, actually, the vast majority do not communicate acoustically, but those that do so by leg wing stridulation, and they have their ears on their abdomen. So that's how you can tell crickets and grasshoppers apart. I did do work with grasshoppers during my postdoc in Germany, but most of my career I have actually spent studying crickets. And among the crickets, I'm going to keep using these terms, the true crickets or the grillets, and the Katydids or the tetygonids. These are the 2, again, major groups of crickets, and we've studied both of them. So what makes them special? Crickets and Katydids, typically males of a species, adult males, produce sound by rubbing their forewings together. And here you can see a tree cricket singing. He has lifted his 2 forewings and he's rubbing them together. And how do they produce sound? They produce sound because one of the veins of the forewing is modified. And if you look on the underside, you can see this row of pegs. I can see the SEM of that beautiful row of pegs. And there's a little plectrum, a hardened part of the wing margin. And basically, he rubs the plectrum of one wing over the file of the other. It's like rubbing a knife across a comb. And that's the basic sound. So one closing movement of the 2 forewings produces a pulse or syllable of sound. So if you look at these pulses of sound, then you pretty much know how often the cricket has opened and closed its wings. And they produce this to attract mates. And one of the species that we've studied quite a bit over the last couple of decades is this little tree cricket called Acanthus henryi. And there's a male over here and a female. Now, females don't have these sound producing structures. They're silent, but they can hear. So when males call typically from a leaf, these crickets live on bushes. You can see the habitat below. And indeed, they have a particular host plant, Hyptis suaviolens, on which they live, sing, lay eggs, and they spend much of their life cycle on these. So adult males typically sing from leaves on these bushes, as you can see over here. And the song of each species is unique. Let me try playing a song for you. I hope you can hear this. It's a very, very familiar sound. Tree crickets are found all over the world. And males will sing for hours after dusk. And if the male is lucky, then a female in the vicinity will hear him and decide to walk or jump and approach him. And if he's even luckier, then she will decide to mate with him. And here you can see a pair that has just mated. The male is standing there with his wings still raised. And they have a very interesting mating, which is that the male basically threads this tiny bag of sperm. He hooks it onto the tip of the abdomen of the female. And the sperm then have to travel from this little bag into the body of the female before fertilization can happen. A female, therefore, has the ability to kick off this bag of sperm. And so the male has to sort of keep her occupied to prevent her from doing so. And he does that by producing a secretion of this gland, the metanotal gland, which is probably nutritious, and she feeds on it for quite a while after mating. And this sort of gives the male the time. It can be half an hour, it can be 45 minutes for the sperm to travel in. So that's the sort of context. The male calls, the female approaches, and then she mates with him. And then you have this transfer of sperm. One of the observations that you should keep in mind, and which is shown over here as the result of an experiment, is that if you put a male and a female tree cricket together and you look for how long the female actually stays mounted on the male before she will kick off the spermatophore, females tend to mate for much longer with larger males than they do with small males. You can ignore the graphs on the right. They all say the same thing. Basically, females will stay mounted on larger males for much longer than they will on smaller males, which means that larger males have a much greater chance of having more of their sperm enter the body of a female. This is something that it'll become apparent why I'm telling you this. So given that females seem to actually prefer these larger males, the question then is, can females actually distinguish between males of the same species based on individual variation in the song features of males? Can they make this evaluation then from a distance and know whom to approach and whom not to approach? Right? In other words, is there female preference for males based on their call features in the wild? So to answer this question, you actually have to do a lot of work. You have to first sort of look at call feature variation between individual males in the wild. You have to look at how consistent these call features are within and between individuals across time, which is often called the repeatability. And you then have to ask whether there's any relation between these call features and any aspect of so-called male quality, for example, size. You then have to look at the female end of things and ask, do females prefer different call features? Are they sensitive to differences in some of these relevant call features? And finally, what are the effects of the ecological context? That is, how animals are distributed in space, and acoustic context, that is the sort of acoustic sound fields. So what are the effects of the ecological context on both signals and preferences? And this is the sort of set of questions that we asked about these animals. And much of this work was done by the spectacular PhD student, Ritik Deb, who actually went out into the field and individually marked something like 200, 300 tree cricket males. And he then would go out night after night into the field and record the songs of these individually marked males. And by this sort of painstaking method, we then had multiple recordings of the same individual males followed over time. Okay? And to just give you a feel for what this tree cricket song that I played to you a little while ago looks, if you visualize it on the screen, this is a recording. And here is what we would call the oscillogram, right? Which basically has time on this axis and song amplitude on that. And you can see that the cricket has a chirping pattern. That was the purr, purr, purr that you hear from the tree cricket. But each sort of chirp actually has substructure in it. And if you look deeper, you will see that a chirp itself is made up of these individual syllables. And each of these syllables actually come from the single wing closing movement that I spoke to you about. Now, the rate at which these syllables are made, the rate at which these chirps are made, tend to be quite species specific, as is the pitch or the carrier frequency of the song. So what you're looking at in this panel is a power spectrum, which shows you at what frequencies most of the energy of the song is concentrated. And in these true crickets, most of it is concentrated in one frequency, which is called the carrier frequency, and this is also species specific. But not only are these species specific, of course, there will be variation from male to male and in all of these different song features, and that is what Hrithik recorded and then characterized. He looked at the individual variation and then he measured something called the repeatability, which is how consistent are these features within males, both within a night, so if you make multiple recordings in a night, and across nights of the same male. Now, repeatability can vary from 0 to 1. So 0 would mean that they're completely inconsistent, whether it's within nights that you're looking at or across nights, and 1 would mean perfectly consistent. What we find is that most of the temporal features, I've lumped them together here, because most of the temporal features, that is the rates of these chirps, the durations, the rates of the syllables, their durations, tend to have relatively low repeatability across nights. That is, if you measure the same individual across nights, they're going to vary quite a bit, fluctuate quite a bit from night to night. They also fluctuate quite a bit within a night. On the other hand, the carrier frequency, the pitch, has high repeatability. This is quite unsurprising because these, the pitch is really determined by biophysical properties of the wings, and those tend not to vary quite as much. And finally, the sound pressure level, or how loud the call is, very interestingly in this species, is very consistent, very highly repeatable within nights and less so across nights. So now, if you think of it from the point of view of a female trying to make a judgment, it's not very useful to look at these temporal features. The carrier frequency is highly repeatable, that is, it's consistent within a male. And so we asked, larger males should have larger forewings. They should then have larger resonating structures or harps, as they're called. And this should result from the physics of the system in lower call frequencies, right? So large males should have lower call frequencies than small males. And maybe females could use this. So males calling at lower frequencies should be preferred by females. So the first thing we then asked is whether this call frequency or carrier frequency is a reliable indicator of tree cricket male body size. And as you can see from this plot, what you have here is an index of male body size combining a number of morphological features. And you can see that larger males on average have lower call frequencies. So yes, larger males have lower frequency songs at a given temperature. You have to remember that these are poikilotherms. And so if you change temperature in these tree crickets, not only their rates of their songs, but their frequencies also change. So everything has to be corrected for temperature. We then did a series of long, tedious behavioral experiments in the lab where we would play back the calls of two different males or two different calls that varied only in the carrier frequency. So they were played back simultaneously to a female. A female was released at this release point that you can see here. She climbed up to the top where she was equidistant from the two speakers, which were at the same intensity at this position, the two calls. And the only thing that was different was the carrier frequency of these two songs. So basically, we were doing a choice experiment to see, does she go towards the lower frequency? What you see on the right are the results of these experiments. Pairwise combinations, I won't go into the details. Each pair is a combination of a lower and a higher frequency within the natural range of variation. The upper panel, they were all done at a lower intensity, 45 dB. The lower panel, they were done at a higher sound intensity. What you can see is that actually none of these paired trials where we measured the number of females that either approached the lower frequency or the higher frequency arm, none of these were significantly different from each other, except one. And in this case, she approached the higher frequency one, not the lower frequency one. Basically, females didn't really care to use this information in our lab experiments, at least. We then went out, we asked, why might this be happening? It could well be that females don't use this information because the tree cricket call carrier frequency actually changes with temperature, which is what is shown in this graph here. And for Echanthus henryi, it's shown for 3 different species. And you can see that the call carrier frequency can vary by as much as a kilohertz over a 10-degree temperature range. And at least on winter nights, you can get that kind of temperature drop even within a night. So possibly this isn't that reliable an indicator. We don't know whether that's right, but that's our speculation. We then looked at another possible indicator, which is sound pressure level or how loud they are. If you remember, the SPL had low across night, but high within night repeatability. And interestingly, it actually had no correlation with male body size. So you can be a small male or a large male, and you could be loud on one night and soft on another. It's possible that the SPL reflects body condition. We have some evidence that it might. So females might go towards louder males because they are in better condition, possibly. When you do a lab experiment in which you, again, have 2 speakers playing out the identical male calls with one louder than the other, playing 50 decibels against 53, or 50 against 56, or 50 against 59, slightly louder, quite loud, and very much louder. The white bars are the higher SPL, and the y-axis tells you how many females went towards, what proportion of females went towards that side. You can see it's a very dramatic result that the females always go towards the louder call, even if the louder call is louder only by 3 decibels, which is quite amazing, okay? Because the energy difference is not that large. So they do always go towards louder calls, but how often do they really hear louder calls in the field? And to test this, Ritik did this very, very nice study where he went out into the field and mapped the positions of individual calling males. And that's what you see in these 3 panels up here. They are examples of 3 choruses. So you know the position of each of these males. He measured how loudly each male was calling at source. And in a series of experiments, he also figured out how these calls attenuate out there in the field. And he used that information together with the hearing threshold of the female, which we knew from other experiments. How loud does a sound have to be for a female to hear it? If you have all of these pieces of information, you can then construct these sort of acoustic perceptual maps. What we have over here, the center of each circle is the position of a calling male in the chorus, and the sort of radius of the circle tells you how far out his call will travel before a female can no longer hear it. So this is his broadcast area, so to speak. And as you can see over here, and I've shown you 3 different examples, 3 different choruses. In many of the choruses, the sound fields of males actually don't overlap. What that means is a female standing in the sound field of one male is very unlikely to hear more than one male at a time. And when you ask how many of them, by also mapping the positions of females in the field, what you see is that 15% of the females, only 15% could hear more than one male at a time, 55% could hear only one male, 30% of the females were not within the hearing range of any calling male. What does this tell us? It tells us that in the field, even though females in a choice situation would go towards a louder male, in the field, they're mostly hearing one male at a time, so they don't really have the opportunity to exercise that preference. Okay? However, there's one situation in which this can change. And that leads me to the next part of my talk. These 3 crickets are quite amazing because they make amplifiers. Yep. So they chew these little holes in the leaf, put their legs through them, and sing with their wings across the hole. And you can see over here, the leaf is so much bigger than the insect. What this does is that this acts as a baffle and amplifies their sound. Let me show you one. Hope you can see this video. And if you ask, how much amplification do you get? That depends upon the size of the leaf. If you take a small leaf, that is among the smaller leaves that you find on this plant, versus a medium-sized leaf versus a large leaf, what you find is that small leaves give you only about 4 to 5 decibels of amplification. Medium-sized leaves give you 6 to 8. And if you really pick large leaves, then you can go up to 10 to 12 decibels, which is quite a bit. You can get 3 times as loud. Okay. And in that situation, then these males that make baffles should have a big advantage. We then asked whether males care about these leaf sizes. And the first thing we did was just observation. And when you look into the field with males who are not calling from baffles and males that are calling from baffles, then what you find that those who are calling from baffles are actually on larger leaves. So they appear to choose larger leaves. We confirmed that in a lab experiment where we brought in males from the field and we offered them either small leaves, medium-sized leaves, or large leaves, or these extra large leaves. These are leaves of plants grown in our nursery, which were larger than found in the wild. And what you can see is the proportion of males that actually made baffles. Almost no one wanted to make these baffles from small leaves, and as leaf size increased, then the probability that a male would actually make this amplifier went up, which tells us that they're somehow able to gauge the size of the leaf and make their decisions accordingly. What's the benefit of doing that? As you can imagine, the volume of your sound field will go up quite a bit. And that's what's shown in this graph. So this, on the x-axis, shows you what the calling SPL of the male was without a baffle. And then you can look at what is the increase that the male gets in the volume of his sound field if he builds a baffle. And to cut a long story short, a baffler can, again, increase the volume of his sound field hugely compared to a non-baffler, which means that females can detect bafflers from much further away, and they're also louder than non-bafflers around them, so they should have an edge in competition. Baffling could be resource limited. What you see in this graph is a frequency distribution of the size of leaves of the host plant that are found in the environment. And if you then superimpose the sizes of leaves on which males were found baffling, then 78% of the leaves sampled in the field were smaller than the median leaf size used for baffling, which suggests that, that leaves that are suitable for baffling may be a scarce resource, and explain why we find very few singing males out in the field who are actually bafflers. Then the next interesting question we asked was, well, who baffles? Who makes these amplifiers? Since only a few males make them. What we found in the field was that those who called from baffles were males whose calling song SPL on that night was lower than average. That is, if I'm not so loud tonight, then I'm more likely to make a baffle, which makes sense. If I'm already loud, then maybe I wouldn't. But what was very, okay, we confirmed this also in lab experiments in which, again, we gave them leaves of different sizes, extra large, large, medium, and small. And we asked whether the males who made the baffles were actually those who were softer callers without the baffles. And as you can see in every case, that is true. Those who didn't make baffles on average had louder calls than those who did, irrespective of leaf size. But what was really intriguing for us was that smaller males also tended to baffle more. So males that were smaller also had a higher probability of baffling than males that were larger. Again, we confirmed that in an experiment, and you can see here that small males on average have a higher probability of baffling than do large males. The white bars are the small males, okay, are the large males. So if we put this together, what it suggests that baffling may be some kind of a cheater strategy. So you make a tool that amplifies your sound. This could be used by less favored males, that is males who are softer in SPL or smaller. May be used by less favored males to gain more matings and increase their chances of paternity. I'm now going to switch gears and move to the cost end of the story. Right? So we've been talking about the benefits of calling and of baffling. Let's talk a little bit about the costs. There are different kinds of costs to make finding, and one of them is, of course, the risk of predation. And it's clear that if you call, you're probably going to be more conspicuous than if you don't. But if you look at how predation risk in the communication context has actually been studied over the years, much of this has been studied in classical systems such as bats and katydids or bats and frogs. Most of the studies are focused on the acoustically orienting predators, unsurprisingly. There are very few studies that actually estimate predation risk in the wild, particularly in systems that are classical models like crickets and katydids for mate choice and sexual selection. Further, most studies conclude that predation pressure is a strong agent of selection on signaling males, but there's a huge bias in the literature. Most people study the signaler and very few study the receiver, the silent receiver. And why is that a problem? So you end up assuming that signaling is riskier than searching silently, but searching also carries risks and costs due to movement. And these are rarely compared directly. Now, the mate finding strategy that an animal should have should be determined by the relative costs of signaling and searching for the two sexes in specific ecological contexts, but there's very little empirical work on this. So that's what we decided to do. We decided, first of all, to figure out who really is the ecologically relevant predator for our tree crickets. And for lack of time, I'm not going to go into how we figured this out. We figured this out using acoustic playbacks of calls in the field to see whether there were acoustic aerial predators such as bats. We then looked at potential terrestrial predators by a whole range of quadrat sampling, who actually lives on these bushes along with the crickets. We did predation experiments both in the field and the lab with these would-be predators. And based on all of these and their abundances, we narrowed down the major predator of this tree cricket to be a green-legged spider. It's a free-moving bright green spider, and spiders larger than 5 millimeters are really the major predators of this tree cricket. So we use these predators for our experiment. I'll just quickly show you spider sort of attacking cricket. That's an unsuccessful one. The cricket jumped off. Let me show you a successful attack. Spider jumps on and catches the cricket. So how did we go about trying to estimate predation risk? Predation is very hard to see in the field. So we sort of broke this down into different probabilities. Since these animals live on these bushes out in the field, the first probability that we try to look at is what's the probability of a cricket co-occurring with a spider on a bush in the field. This was, of course, observational, done in the field. And then if you do co-occur with a spider on the bush, what's the probability that the cricket and the spider will actually encounter each other within a period of 1 hour or 2 hours in the peak calling time? And finally, what proportion of spider attacks actually end up being successful? So if a spider jumps at me, how often do I escape? How often do I get eaten? And we estimated these 3 different probabilities, the first by doing observations in the field, the second with a setup where we had the bush and we had spiders and crickets on the same bush, and we asked how often do they actually encounter each other? And finally, how often does a spider manage to catch a cricket? And we did this with 4 different treatments. We did this with calling males. We did this with males that were silent, not calling on a bush. We did this with females that were actually trying to find males, and we got them to do this by doing playbacks on a bush so that a female would move through the bush on which a spider was present to try and find a calling male. And of course, the control females who were silent. And if we, that's for the experiments. The first probability, of course, was simply the chance of co-occurrence of a male or female on a bush with a spider. This is a very busy slide. Let me go through this. So the top panel is the chance of co-occurrence of a spider and a cricket on a bush. For each of these 4 treatments, a calling male, a non-calling male, a female, and a female not doing phonotaxis. What we see is that on average, the median value here is about 0.2, 20% chance of co-occurrence on a bush, and it's not different between these 4 categories. Okay. The second panel gives you the result of the second experiment. What's the chance of encounter given co-occurrence? Again, the median value ranges there from about 20 to 35% chance of encounter. And again, it's not significantly different between these 4 treatments. And finally, what's the chance of mortality if a spider attacks you? It's less than 10%. And again, it's not significantly different between the treatments. This was a completely unexpected result. What we found was no significant differences, basically, in predation risk between the signaling males and the searching females, and indeed between them and the controls. Okay, so what does that say? If we now calculate the overall risk of mortality by multiplying these 3 probabilities, then again, we find no significant differences between the 4 treatments. And we can actually put numbers to some of these. And you can see that these numbers on a per night basis are fairly low. But what's more important is that they're not different between the treatments. So what do we conclude from this? At least in this system, with signaling males and searching females and an ecologically relevant predator, they face similar levels of predation risk. And also the cost of acoustic communication in terms of predation risk in this system, at least, seems to be negligible. So that's what we learned by looking at the truth. And when we tried to publish those results, of course, we had a lot of difficulty because nobody really believed the results with the tree crickets. And one of the big criticisms we got there was, oh, spiders don't hear, you're using the wrong predator. But you know, it's the ecologically relevant predator. So we feel quite justified in using them. However, there is another system, which is between predatory bats and calling katydids. Again, katydid males call for females, and bats do eat them. And there's been decades of work on this sort of canonical predator-prey system. We had also done a lot of work in the tropical evergreen forests of Kudremukh National Park in the Western Ghats in India, where we had, again, over a couple of decades, characterized the entire acoustic community of that area of tropical evergreen forest. Is a different story. We were looking at masking interference and how they managed to communicate in noise. Quickly, I know I'm running out. That's a canopy katydid, an understory caller. I'll give you some of these nice ones. A katydid in the leaf litter. All these live in the same patch of forest. This is a tree weta. The cut, cut, cut sound that you hear. So there's this sort of community of 20 species of crickets and katydids. And after sort of years of misnetting in these forests, we found that the community of bats also consists of about 20 species. And by a process of elimination that I won't go into, we identified who were the major bat predators in the system. This is the lesser false vampire. It's not a vampire, Megaderma spasma. It's an insectivorous bat. If you go into a roost of spasma, what you will see on the ground is piles and piles of these discarded insect wings and ovipositors and legs. And as you can see from a lot of these green wings, they eat a lot of katydids. So we followed 5 of these roosts through a year. And by picking up all these culled remains, which the bats reject before eating, we could count up, how many remains there are of different orders of insects. And the red ones here show you the Orthoptera, of which 98% are katydids or tetygonids. So anywhere from about 40 to 80% of their diet consists of katydids. So they do eat a lot of katydids. But here came the first big surprise. Because we can tell by looking at the wings of katydids whether they are males or females, because the sound-producing structures are on the wings, and only males have them. We could also figure out whether male katydids were preyed upon more heavily than females. Remember, males call, females are silent. And here's where we got our first surprise. We found twice as many female katydid remains as we did of males. And this was something that needed explanation. And so we set up behavioral experiments in enclosures with the bat predators to test how often bats would approach the songs of these male calling katydids versus how often they would approach live tethered females in which we induced flight. Let me show you a video of one of the playback experiments. This is shot with infrared camera in an enclosure. We've got a loudspeaker over here playing back the call of a katydid. There is an immobilized katydid on the leaf, and there's a bat somewhere in the cage. I'll tell you. So this is all background noise. And I'll tell you when the call, that's when the call of this katydid, it plays from only one of these two. Playing again. Now, And I hope you saw the bat down to it. So we did such playback experiments, and we also did experiments with tethered insects in flight. And we looked at the number of times bats would come and approach them. To summarize it, for 3 very different kinds of katydid calls, there was approximately a 1 in 3 chance that a bat would approach it within 3 minutes. But if you induce flight, bats will approach immediately 100% of the time. In other words, flying females are 2 to 3 times more likely to be approached by bats than calling males, all else being equal. So this sort of corroborated what we were seeing in the diet of these bats. I think, can someone tell me if I'm out of time? I'm almost at the end.
You have just about 5-ish minutes left.
5 minutes, yeah, perfect.
Thanks.
So given that, we have these calling males who might be approached, and we have females for whom flight is so dangerous. In one of these canopy katydids, we actually asked, well, what do females do if flying is so risky? So we tried these very, canonical Phonotaxis experiments with this canopy katydid species, where we had a speaker which played out the call of the male of her species. And what any self-respecting cricket will do is walk down to this tree and approach the speaker, maybe fly to it. I'm going to show you. So the female is at this end of the T, the speaker is over there in the next video, and I'm going to show you what she does. So you can see the female standing on the leaf, and you will hear the playback of the male now. Watch the leaf carefully. Now you can see it's beginning to shake that leaf.
Recording in
fights
progress.
when two males encounter each other. And as far as I know, at least with the true crickets, whatever I have worked on and seen so far, very often it's the the call is given out at the end of a fight by the victor.
Oh, so it's I guess it's not a dominance thing; it's a submissive thing then.
Yeah, I don't know. This is what I've often seen with true crickets, and that seems to intimidate the loser. Yeah.
Interesting. Okay. Yeah, we're back on the live stream. Great. So, so the second question, also from Saeed, says, awesome. Does sound production in males relate to body size? Is body size frequency dependent, which is something that we do see in fish?
You mean is frequency body size dependent?
Yes. Yes, is frequency body size dependent?
Yes, but definitely. Both with the tree crickets and with the field crickets, body size is related to frequency, and I mean within species. Yeah. So as I showed you in the talk, larger males have lower frequency sounds, but there is a bit of a catch with the tree crickets because tree cricket carrier frequency can change with temperature. Okay? So the relationship is there, but it depends on the temperature. With field crickets and katydids, it's less so. And yes, larger males will have lower frequency sounds, but as I showed, and we have tested this with both tree and field crickets, females appear not to care about it. It's interesting too.
Interesting, thank you. And for, I guess, because we did just start the live stream, I'll sort of repeat Saeed's question from before, who was asking if crickets produce sound only to attract females or if it is antagonistic. And Rohini had mentioned that you do notice some antagonistic sounds, but you did mention that usually the loser is the one that makes the sound, right? The one that
the victor.
Oh, the victor. Oh, sorry, I thought you had said the victim. Okay, yeah. So it's the one who wins the male antagonistic interaction that makes the sound.
Yes.
Interesting. Okay, and then so we will move on to another question by Erica Marquis de Santana, who says, "Great presentation. Is there individual variation in female preference for male calls? And if so, is it related to other female traits such as size, age, or condition?"
Is not something I have tested in these crickets, repeatability of female preferences. Lots of other people have worked on this stuff. And I remember from 30 years ago, I worked on this for grasshopper female preferences. And grasshopper female preferences, yes, there's huge individual variation. There's individual variation in which features females, individual females weight different features. And they can be quite consistent over time. And some females are very non-choosy, other females are choosy. And at least in the grasshoppers we studied, again, they were quite consistent over time. So there were females who, from the day of their birth to death, were highly selective, and there were females who changed their preferences, and there were females who were non-choosy, beginning to end. And I would be willing to guess, but I would find the same thing with crickets.
Thank you. Thanks. So
I like to say they're just human, but
yeah, they're similar humans. So Natasha Rossi says, "Amazing research, and thank you so much for your talk, Professor Balakrishnan." And then we do have one more question from Cassandra Aymon. So great talk, and thank you, Rohini. You said that females reduce mating time when mating with smaller males. So would it be possible for the female to feel the male body size when mating and adjust accordingly?
Fantastic question. We don't know the answer. We don't know how females figure out male body size. Is it, maybe there's a chemical cue somewhere which she can use? I mean, but, I can speculate. We don't know the answer.
Is it a research in progress? Is that a sort of, because you said good question, does that mean it's a topic you're currently considering?
No, I think it's a very hard one. It's a very hard one. One might be able to do some smart behavioral experiments, I know, but it's very clear that they can tell. Now, it's also clear that they don't care to use any of the acoustic information that they might have to make that choice. So basically what I'm saying is they'll approach any male, they will mate with almost every male they approach, but they do calibrate this time. Do they know? I don't know. Do they know by touching, from contact pheromones, from cuticular hydrocarbons? Do they figure out when they start drinking that metanotal gland secretion? I think the answers lie there, and I'm just not enough of a chemical ecologist to sort of go down that path.
A lot of different options. Okay, great. So that is actually the end of our